So this gentleman here is Friedrich Miescher, and he isolates DNA for the first time from pus, and pus contains a lot of cells that have nuclei, so nuclei contain DNA. So that's where he gets that from. They had a big fascination with pus because it was easily available back then, not so many antibiotics. This guy, Walter Fleming, describes mitosis. So we would not be able to grow if our cells didn't divide and become 2 and then 4 and then 8 and so on and so on. So he describes this process of cells dividing. And that's that again. So this gentleman, Valentin Hacker, that's probably German, I'm not saying that right, he first used the word stem cell to describe a cell in a crustacean like a, I forget it, never mind, I won't even try. So he saw that one cell became many different types of cells. So he was the first one to use the word stem cell. So that's not him. This guy, Hans Spemann, published the results of a transfer where he took a nucleus from one cell and put it in another and watched something happen, watched things change. So this would be considered the first cloning. He was not able to create a whole new salamander, but he was able to start something anyway. So that's 1938. Okay, so we've got two guys named Avery and McLeod, and there's one more fellow, I couldn't find his picture. They show that DNA can change the property of cells. So if we change the DNA in that nucleus, we can change how that cell acts. So, and actually, if you think about it, all the cells, let's say on your pointer finger, actually have all the same DNA as the cells in your stomach. And it has all the same DNA as the cells in your eyes, but they're not read the same way. Each cell is not reading the same recipe, all right? So, which is good because we don't want the cells in our eyes making the hydrochloric acid, for the stomach, because it would just dissolve our eyes, right? So this is important that a cell can discriminate what bit of DNA they read. But we can also exploit that and change a cell by changing its DNA. Okay, I don't know if you can see that picture very well. This is, we all know what Watson and Crick did, right? Does anybody know what Watson and Crick did? Somebody under 18. Do they know what Watson and Crick did? Yeah, so they discovered the shape of DNA. So it's a double helix. So that's what that picture is, the model of the double helix. The names in red mean they won a Nobel Prize, right? Okay, this guy, Nierenberg, he figures out that DNA actually has a very specific code for specific, they're not proteins, they're 4 specific molecules that determine when you put them in groups, they become the amino acids which can become proteins. Okay, so we're on to the second slide. So these guys actually were the first ones to perform first successful bone marrow transplants. So hopefully this should not come to you as a surprise that bone marrow transplants are actually the first example of using stem cells for treatment. So this has been 40 years worth of work altogether. So Dr. Thomas, who is on the right actually, he won a Nobel Prize for his work. This thing is not Leroy Stevens. This is what he found, or something like what he found. So what he found was he was studying mice and he saw this strange tumor growing in the testicle. So he pulled it out, and this tumor had hair and teeth and bone and muscle, and it had everything. And how could that be? But it didn't look like a mouse. It just looked like this thing with all those pieces. And this is called a teratoma. So this gave him the idea that maybe there is a particular cell that can divide and become all these different things. So this was actually quite an important discovery. And maybe you've met someone who's had a tumor where they pull it out and it has hair. So I don't know if you can see the little hair sticking out. This one had golden hair. It could be brown hair, it could be whatever. So I met someone down in St. Louis and standing at the arch, and she starts telling me about her surgery, and she had this tumor removed from her abdomen, and she goes, "It was my twin." And I was like, boy, that sounds like I heard that in a movie one time, but she had a teratoma. So I don't know that it was her twin. It was just this. So these two guys became very, very rich. This is Cohen and Boyer, and what they did was they were able to take a piece of DNA, put it in a bacteria, and make that bacteria produce insulin, which is the drug you need when your pancreas is not working properly. So when you have type 1 diabetes, you might need to take insulin. And so it was really hard to isolate insulin all the time. So what if we had something that could just make insulin? So they were able to grow lots of bacteria that produce lots of insulin in giant vats, probably as big as this room. So these guys actually became very rich doing this. They made an entire whole company that did this. So this was really, really important. So this is our first sampling, putting DNA in something and making that organism do something completely different. Bacteria do not normally create insulin. Humans do, mammals do.